Hi students, this is Mr. Eckert. We're going to go through Target H12, which says that you should be able to describe the factors that result in genetic variation, and that would include mutations, something called crossing over, independent assortment, and sexual reproduction. So first, why is genetic variation so important? Well, first of all, just think about what the word genetic variation means. Variation, of course, is differences. And genetic refers to genes, so that would be differences in DNA and genes, and that is a key factor in the evolution process. Now, we're going to define evolution as changes within populations of living organisms. Most students and even adults have the misconception that an individual organism, like an individual ladybug here, would be able to adapt to a change in its environment. And they don't, because they can't change their DNA. But a population of living organisms, because it has genetic variations, of course, all those ladybugs look pretty much alike to me, but genetically they're different because they sexually reproduce. So, in fact, you can even see where this ladybug right here, its spots are a little bit less pronounced on it. So that is a source, or that is an example, I should say, of genetic variation. And because of genetic variation, when there is a change in the environment, then a species will have a better chance of surviving. Think about if all of these ladybugs had the exact same DNA and you sprayed them with a pesticide that killed one. Well, it's going to kill them all. So the fact that there's genetic variation is a positive for any species. So remember, genes, most of them have at least two forms or alleles. A different form of a gene is an allele. And all organisms have genetic variation then because they receive at least two different alleles for each trait from each of their parents. So lots of individuals are heterozygous for genes. They receive one allele from one parent and a different allele from the other. A population is a group of individuals of the same species that can breed with each other, and it doesn't say it here, but they must be able to produce viable offspring. Their offspring must be able to reproduce. So when we use the term gene pool, we're talking about all the genes, including all those different alleles that are present or found in a population of living things. We can measure the relative frequency of an allele, and that would be the number of times that it shows up in a gene pool compared to all the other alleles. So for example, in these flowers, we can measure the frequency of the big R allele, which codes for red, in the total population of flowers. And it might turn out to be something like 60%, let's say. So, as it says here, relative frequency then is often expressed as a percentage, but is not related to whether an allele is dominant or recessive. Remember, we've probably talked about in class how 5 fingers is actually a recessive allele, and yet, of course, that's the dominant phenotype in humans. So when scientists, it says, determine whether a population is evolving, they're going to look at probably the gene pool, because if there are changes in the gene pool, then that would be evidence of evolution. So you can see here a sample population of mice might have a certain percentage of the allele for brown fur and a certain percentage of the allele for black fur. So if you can measure that and then come back later at another time, measure it again. If there was a change in that, then that might indicate evolution. So in genetic terms, as it says, we're going to define evolution then as a change in the relative frequency of alleles in a population. You can see here these two hummingbirds have very different bill lengths. And so that's determined, of course, by their DNA. Once again, if we look at that percentage and see changes in that over time, that would indicate evolution. So what are some sources of genetic variation? Well, the main sources of genetic variation are mutations, random mutations that would occur in the DNA. That would be gene mutations or chromosomal mutations, and the genetic shuffling that results from sexual reproduction. So a mutation is a change in a sequence of DNA, and they occur because of mistakes, maybe when DNA replicates, or as a result of what are called mutagens. Mutagens are anything that can cause a mutation in DNA. It might be UV radiation. It might be a certain chemical like benzene, which is a known chemical that's in tobacco when they treat tobacco. You can see this fruit fly on the left is a normal fruit fly, and you can see right here are its antennae. They almost look like little sparse feathers coming out of its head. And here's a fruit fly with a obvious mutation. Instead of the antenna, it has a leg that's growing out of the front of its face, and you can see how that would be a terrible disadvantage for that fruit fly. But even though this is an example of a mutation that affects a phenotype, they don't always affect an organism's phenotypes. So gene shuffling is the cause of most differences in organisms. Most phenotypes, you can see in this population of humans, that you've got obvious differences between them, and that's due to gene shuffling. So how do genes get shuffled then? Well, sexual reproduction results in gene shuffling for the very fact that which sperm gets to fertilize the egg is a totally random event. There might be hundreds of millions of sperm that are vying for that opportunity to fertilize the egg, but only one, as you know, is going to get that opportunity. So that's a random event. So that results in genetic shuffling. Independent assortment of alleles that occurs during the process of meiosis, the formation of sex cells, also results in gene shuffling, much like shuffling a deck of cards and dealing them out is a random event. Well, when the sex cells are formed, the alleles for each trait are shuffled in much the same way and dealt out to the different sex cells. So it says each pair of alleles segregates independently of other pairs of alleles during that formation of gametes. That means that you inherit each trait independently of one another. For example, what this is saying is it's possible you could get your mom's hair color and not get her eye color, right? Because you inherit those alleles independently of one another. They're not connected to each other. If they were, then let's say if your mom had blue eyes and red hair, and you inherited her blue eyes, then you would automatically get her red hair. So this diagram shows what I was talking about. You can see alleles on chromosomes here in the parent cell, and during the process of meiosis you can see how those alleles get shuffled. These are the gametes that form, and you can see how they shuffle just randomly, just like cards. So that's the law of independent assortment. Here you can see the same shuffling of alleles into individuals in a Punnett square. So another source is crossing over. Remember, during prophase 1 of meiosis, homologous chromosomes pair up and form tetrads, and they can exchange actual pieces or segments of those chromosomes, and that's called crossing over. So that also results in genetic variation, or recombination as it's also sometimes called. So you can see in this diagram, again, we have one chromosome from mom, one chromosome from dad. Crossing over takes place, pieces exchange and reattach, and so that is why it's called recombination, and results in gene shuffling. So that's it for that target. Now, I will say at the end of this target here, which again was H12, that target H13 is on the history of our understanding of genetics, and rather than go through PowerPoint and notes, you're going to do a online investigation of the different individuals that are critical to our understanding of heredity. So no PowerPoint notes for H13. It's an online assignment that you'll do.